Dolly, sheep. Dolly, 5 July 1996 to 14 February 2003, was a female Finnish Dorset sheep and the first mammal cloned from an adult somatic cell. She was cloned by associates of the Roslin Institute in Scotland using the process of nuclear transfer from a cell taken from a mammary gland. Her cloning proved that a cloned organism could be produced from a mature cell from a specific body part. Contrary to popular belief, she was not the first animal to be cloned. The employment of adult somatic cells in lieu of embryonic stem cells for cloning emerged from the foundational work of John Gordon, who cloned African clawed frogs in 1958 with this approach. The successful cloning of Dolly led to widespread advancements within stem cell research, including the discovery of induced pluripotent stem cells. Dolly lived at the Roslin Institute throughout her life and produced several lambs. She was euthanized at the age of 6 years due to a progressive lung disease. No cause which linked the disease to her cloning was found. Dolly's body was preserved and donated by the Roslin Institute in Scotland to the National Museum of Scotland, where it has been regularly exhibited since 2003. Genesis. Dolly was cloned by Keith Campbell, Ian Wilmot, and colleagues at the Roslin Institute, part of the University of Edinburgh, Scotland, and the biotech company PPL Therapeutics, based near Edinburgh. The funding for Dolly's cloning was provided by PPL Therapeutics and the Ministry of Agriculture. She was born on 5 July 1996 and died on 14 February 2003 from a progressive lung disease that was considered unrelated to her being a clone. She has been called the world's most famous sheep by sources including BBC News and Scientific American. The cell used as the donor for the cloning of Dolly was taken from a mammary gland, and the production of a healthy clone, therefore, proved that a cell taken from a specific body part could recreate a whole individual. On Dolly's name, Wilmot stated, "Dolly is derived from a mammary gland cell, and we couldn't think of a more impressive pair of glands than Dolly Parton's." Birth. Dolly was born on 5 July 1996 and had 3 mothers. One provided the egg, another the DNA, and a third carried the cloned embryo to term. She was created using the technique of somatic cell nuclear transfer, where the cell nucleus from an adult cell is transferred to an unfertilized oocyte, developing egg cell that has had its cell nucleus removed. The hybrid cell is then stimulated to divide by an electric shock, and when it develops into a blastocyst, it is implanted in a surrogate mother. Dolly was the first clone produced from a cell taken from an adult mammal. The production of Dolly showed that genes in the nucleus of such a mature differentiated somatic cell are still capable of reverting to an embryonic totipotent state, creating a cell that can then go on to develop into any part of an animal. Dolly's existence was announced to the public on 22 February 1997. It gained much attention in the media. A commercial with Scottish scientists playing with the sheep was aired on TV, and a special report in Time magazine featured Dolly. Science featured Dolly as the breakthrough of the year. Even though Dolly was not the first animal cloned, she received media attention because she was the first cloned from an adult cell. Life. Dolly lived her entire life at the Roslin Institute in Midlothian. There she was bred with a Welsh mountain ram and produced 6 lambs in total. Her first lamb, named Bonnie, was born in April 1998. The next year, Dolly produced twin lambs, Sally and Rosie, and she gave birth to triplets, Lucy, Darcy, and Cotton, in 2000. In late 2001, at the age of 4, Dolly developed arthritis and began to walk stiffly. This was treated with anti-inflammatory drugs. Death. On 14 February 2003, Dolly was euthanized because she had a progressive lung disease and severe arthritis. A Finn Dorset, such as Dolly, has a life expectancy of around 11 to 12 years, but Dolly lived 6.5 years. A post-mortem examination showed that she had a form of lung cancer called ovine pulmonary adenocarcinoma, also known as Jagdsekje, which is a fairly common disease of sheep and is caused by the retrovirus JSRV. Roslin scientists stated that they did not think there was a connection with Dolly being a clone, and that other sheep in the same flock had died of the same disease. Such lung diseases are a particular danger for sheep kept indoors, and Dolly had to sleep inside for security reasons. Some in the press speculated that a contributing factor to Dolly's death was that she could have been born with a genetic age of 6 years, the same age as the sheep from which she was cloned. One basis for the idea was the finding that Dolly's telomeres were short, which is typically a result of the aging process. The Roslin Institute stated that intensive health screening did not reveal any abnormalities in Dolly that could have come from advanced aging. In 2016, scientists reported no defects in 13 cloned sheep, including 4 from the same cell line as Dolly. The first study to review the long-term health outcomes of cloning, the authors found no evidence of late-onset, non-communicable diseases, other than some minor examples of osteoarthritis, and concluded, "We could find no evidence, therefore, of a detrimental long-term effect of cloning by SCNT on the health of aged offspring among our cohort." After her death, Dolly's body was preserved via taxidermy and is currently on display at the National Museum of Scotland in Edinburgh. Legacy. After cloning was successfully demonstrated through the production of Dolly, many other large mammals were cloned, including pigs, deer, horses, and bulls. The attempt to clone Argolai, mountain sheep, did not produce viable embryos. The attempt to clone a Banteng bull was more successful, as were the attempts to clone a mouflon, a form of wild sheep, both resulting in viable offspring. The reprogramming process that cells need to go through during cloning is not perfect, and embryos produced by nuclear transfer often show abnormal development. Making cloned mammals was highly inefficient. In 1996, Dolly was the only lamb that survived to adulthood from 277 attempts. By 2014, Chinese scientists were reported to have 70 to 80% success rates cloning pigs, and in 2016, a Korean company, Suam Biotech, was producing 500 cloned embryos a day. Wilmot, who led the team that created Dolly, announced in 2007 that the nuclear transfer technique may never be sufficiently efficient for use in humans. Cloning may have uses in preserving endangered species, and may become a viable tool for reviving extinct species. In January 2009, scientists from the Center of Food Technology and Research of Aragon in northern Spain announced the cloning of the Pyrenean ibex, a form of wild mountain goat, which was officially declared extinct in 2000. Although the newborn ibex died shortly after birth due to physical defects in its lungs, it is the first time an extinct animal has been cloned, and may open doors for saving endangered and newly extinct species by resurrecting them from frozen tissue. In July 2016, 4 identical clones of Dolly, Daisy, Debbie, Diana, and Denise, were alive and healthy at 9 years old. Scientific American concluded in 2016 that the main legacy of Dolly has not been cloning of animals, but in advances into stem cell research. After Dolly, researchers realized that ordinary cells could be reprogrammed to induce pluripotent stem cells, which can be grown into any tissue. The first successful cloning of a primate species was reported in January 2018, using the same method which produced Dolly. 2 identical clones of a macaque monkey, Zhongzhong and Huahua, were created by researchers in China and were born in late 2017. In January 2019, scientists in China reported the creation of 5 identical cloned gene-edited monkeys, again using this method and the gene-editing CRISPR-Cas9 technique, allegedly used by He Zhonkui in creating the first ever gene-modified human babies, Lulu and Nana. The monkey clones were made in order to study several medical diseases.